Thank you for the opportunity to be here. This will be a good complement to everything else we're here today, because I am going to really try to dig in and speak broadly about mosquitoes and slowly address West Nile. I think the lesson here is we really need to understand these systems and the biology of how viruses are maintained. Janet's already talked about that. The point is we need to have a really precise intervention that's going to be appropriate for the system. All right, so speaking broadly, there's indeed more than one species of mosquito in the world. I come across that kind of misconception quite a bit. And so broadly speaking, there are over 3,000 species of mosquitoes in the world. They all are in this member. They're a family, Culicidae, so they're a fly within the order called Diptera. They occur on every continent except for Antarctica. They are considered the world's most important arthropod affecting human and animal health. So arthropod referring to all insects, ticks, spiders, etc. So they are globally extremely important. And it turns out, if we think of some of these really big vector-borne diseases around the world, they tend to be transmitted by mosquitoes that do really well in the urban or the human environment. So in a lot of cases, those mosquitoes have essentially become domesticated. And that's actually true for West Nile virus, malaria, dengue virus. Those are all driven by vectors that really thrive in the human landscape. And then, of course, for these reasons, a lot of resources are used to reduce mosquito populations, not just for these vector-borne diseases, but also separately for the nuisance reasons alone. All right, so here in the United States, there are 172 species of mosquito falling into 14 different genera. So again, we're talking order, Diptera, family, Culicidae, and then the taxonomic unit of genus would be another grouping factor, and then within these would be the species level. So from a public health perspective, this might be an overwhelming list, but the reality is we only are interested in the ones that could potentially feed our, say, humans, or would be involved in the transmission of some kind of vector-borne disease. And so for that reason, we can really actually narrow this down to a few genera that are actually going to be somehow involved in some kind of human health implications. And then here in Texas is large, a lot of diverse landscape. So we have 85 species, 11 genera. There's a good resource, AgriLife Extension has a resource documenting every species in the state and even showing maps of where those species have been detected, in which county. All right, so just so we're all on the same page with just simply morphology, this is the adult mosquito. So it is an insect. It has 3 body segments. It has 3 pairs of legs and 2 wings. And there are actually a lot of other members of the Dipteran family, other families that look very closely, have similar appearances as mosquitoes. So a lot of people will mistake midges, members of this Chironomidae family, or Capulidae, or crane flies. I think a lot of people might see those and think they're mosquitoes. One thing that can be done if you want to specifically look and detect and confirm a Culicidae family or a mosquito would be the presence of this proboscis. That would be the mouth part designed to probe either getting blood meal or getting sugar sources. And then broadly speaking, we've already talked about some of the different mosquitoes and the banding patterns they have. Those banding patterns are present because of the scales. Mosquitoes have little scales in them that are similar to butterfly scales, and it's those scales that create patterns, and that's partly what we use to identify mosquitoes. I just wanted to throw this mosquito out there. This doesn't occur in the United States, but the point is these are scales that are sometimes iridescent in colorations. We do have some really similar mosquitoes that do have iridescent scales, even some right here in Texas. So this is kind of the charismatic megafauna of the mosquito world. All right, so again, looking at this adult morphology, it's actually not that hard to look at these mosquitoes and get them down to, say, the genus level, and a lot of times even the species level. There are certain stop characteristics that even with the naked eye, when you slap a mosquito, you can literally, if you know what you're looking for, you can detect and pick up on these subtle differences. I talked about this proboscis. Mosquitoes, all mosquitoes also have a palate. This is a sensory organ, and for Anopheles mosquitoes, which we've talked about, they are the vectors of malaria. Their palps are very long and similar length to the proboscis. So that's very different from all other non-Anopheles mosquitoes. So that's sometimes if you were to go through a key, a taxonomic key, that would be one of the first things that would separate a lot of different mosquitoes. In addition to that, sometimes we refer to the terminal end of the abdomen. This is their rear end, and whether this ends in a blunt shape or a pointed shape group, two different large groups of mosquitoes. And again, we've heard about Culex mosquitoes. The females would have short palps and blunt abdomens, and they have golden scales, and so immediately we start that to that genus, and you can literally do that with your naked eye. All right. The other thing that's important is when we're, say, processing these mosquitoes or testing them for pathogens, we want to be able to distinguish males from females. Males do not ever take a blood meal. They do feed on sugar sources, though. And so if we're testing these mosquitoes, we don't want to include a bunch of males in with the females, because then that could influence our results. And so male mosquitoes can be distinguished through a couple of reasons. One of the most prominent would be this palp that I'm referring to. In females, they are short or sometimes long, but in males, they're always long and they curve and they have a lot of hair associated with them. So here in the Aedes aegypti mosquito, the one responsible for dengue, you can see that long curved palp. Here are Culex vascatus, the mosquito responsible for what's now here in Dallas. You can see here this long curved palp. And so again, once you can recognize that, that is easy to just quickly distinguish males from females. And now, briefly looking at the larval, the immature stage, there's a lot of features that allow us to uniquely identify larvae. All larvae are aquatic, and they all have to breathe air. And so we've already talked about reasons why in some environments, if there's wave action, that prevents these mosquitoes from contacting the surface of the water with the respiratory siphon. So they do need to breathe air. And that's why oils prevent that contact with air. So that's why those work. Actually, when you identify larvae, many times you can identify larvae down to species level with a lot of confidence, whereas sometimes the adult stages are really hard to distinguish morphologically. And so that's why it is important sometimes to be able to identify these. So we've already seen a diagram of the mosquito life cycle, so this will kind of be a review. They have their complete metamorphosis. They go from an egg to an immature larval stage. They can then turn into a pupae, and then they emerge as an adult. We have heard now that mosquitoes have kind of two general types of egg-laying behavior. Some species of mosquitoes will lay their eggs as a raft. On the surface of the water, there might be 100 or 200 individual eggs in an egg raft. Whereas other species of mosquitoes, probably the majority of the species, would lay their eggs individually. Often those eggs are laid in moist soil or on the edges of containers, and they're laid individually, and they're intended to wait for water to inundate those eggs, and that's going to then trigger hatching. So that's why a lot of times these individual eggs are a synchronous type of hatching event. So after a flood, then we see a huge emergence. Whereas we just heard by Janet, Culex mosquitoes would be asynchronous. They're just going to constantly be producing, and we're going to see a lot of these different stages all in the same environment. So then, once the eggs hatch, they go through 4 larval instars as they're growing, feeding on different microorganisms, bacteria, protozoan, fungi, algae, etc. Once they reach that 4th instar stage, turn into a pupae. Sometimes pupae are called tumblers. Both larvae and pupae have anti-predator behavior, so that's why when we approach them, we see them drop down. They're essentially trying to avoid predation. And then they emerge as an adult. Adult mosquitoes can stay alive for anywhere from a few weeks to a few months. Yeah?
How long do those mosquitoes usually live
Yeah. So dormancy, that actually leads me to the kind of the next concept here. Some of these eggs can remain dormant for a long time, over a year even. However, the Culex mosquitoes, they lay them on the surface of water. Those are going to take a day or two to hatch. And so those hatch really quickly, and then that starts the life cycle. But these mosquitoes that lay their eggs, say in soil that's moist, waiting for water, they can remain dormant for a long time. So in fact, I was going to talk about the different ways mosquitoes overwinter. I'm coming from Michigan where we have this thing called a winter. Down here, there still is some overwintering going on. But in some cases, clearly there's no mosquito activity for several months of the year, but mosquitoes can still manage that. They can still overwinter. Different species overwinter through different ways. And so some species overwinter with dormant eggs. Others overwinter as larval stages. And then for Culex mosquitoes, they overwinter as the adult stage. And so there's certainly going to be different strategies. So these Culex mosquitoes are going to seek underground catch basins, storm tunnels, and naturally they're going to find rock, crevasses or hollow logs, etc. A sheltered environment where they can then be sheltered from colder conditions. All right, now a little more about mosquito biology. Just emphasizing the point that all adult male and female mosquitoes do obtain sugar resources. And so this is probably not appreciated all that often. Sugar sources could come from nectar and flowers, but often also associated with fruit or different kind of berries that might drop to the ground. I know I've encountered a lot of mosquitoes feeding on mulberries or in Puerto Rico, the different kind of fruits that might drop to the ground. Males and females would be attracted to that. Females of most species consume blood, so there are a number of species that just are exclusively sugar-feeding mosquitoes. They do not feed on blood. And we already saw, we referred to Toxorhynchides as a genus of, actually the largest genus of species in the world. They occur in the southern United States. Look at this curved proboscis. That is not intended to probe into a host. That's intended to obtain sugar from a flower or other sources. And so we already heard about how that particular species has been used as a biological control agent. They have a huge larvae, and it's a predatory larvae. It feeds on other mosquitoes. So that's kind of a unique mosquito there. And of the species of mosquitoes that do feed on blood, there are sometimes different subspecies or even in different individual females that can still produce eggs without a blood meal. So that concept is called autogeny, when a mosquito is able to produce eggs without a blood meal. So that can occur as well. And then I wanted to talk about this term called a gonotrophic cycle or gonotrophic period. That is essentially the time from a female mosquito when she emerges. She takes a blood meal. She's going to digest the blood meal, produce eggs, lay those eggs, and then she's ready to take another blood meal. So that period is called the gonotrophic period. So a female mosquito, she can go through several of those gonotrophic periods over the course of her lifetime. And for Culex mosquitoes, that gonotrophic period might be around 6, 7, 8 days is the interval between these blood-feeding events. And once we start talking about vector-borne diseases, that's important because for many of these pathogens, they're not vertically transmitted, meaning the progeny, the mosquito larvae, they're born uninfected. So they have to pick up the pathogen during that initial or at least a blood-feeding event. And then eventually, once the pathogen is now infectious in that mosquito, during a later gonotrophic period, then they're able to reinfect the new host. So here's a common misconception I encounter a lot in the field. Most of my research on West Coast for the last 8 years was in Chicago, just a different, large urban environment, but in a lot of ways really similar to Dallas. So I come across this a lot. A lot of people often associate mosquitoes with rain, and in a lot of ways that's true. We notice those big emergences of floodwater species mosquitoes, and they're a nuisance that we notice them feeding on us. But then often people make that association with West Nile virus. They assume that more rain equals more mosquitoes equals more West Nile virus, but that's not necessarily true. And so one of these main concepts for that reasoning is that we do have these different mosquitoes, and they have different natural histories. Some fall into these categories of floodwater species that I've talked about, where they then have synchronous response. They all emerge at the same time, whereas others would be container breeding mosquitoes. And these container breeding mosquitoes, they can actually thrive in really dry environments. They want stagnant water. And so in Chicago, there's no such thing as too dry for Culex mosquitoes. The drier it is, actually, almost the better it is. Because if you have large rain events, that's going to flush those larvae out of these container habitats. So these are two general types of breeding. So then the point is, which type of mosquito is the vector or is responsible for transmitting the pathogen? So for West Nile virus, it's primarily container breeding mosquitoes. But as an example, for other vector-borne diseases, mosquito-borne diseases, say Rift Valley fever, that is primarily amplified by a floodwater species. That's in Africa only. It's not occurring here in the U.S. So large epidemics of Rift Valley fever virus occur after a large rain event. And so, again, two totally different types of systems. All right, so now talking about pathogens. The point here is, why is a mosquito a good vector? What's the biology behind that? So the point is, we've already heard about, from an insecticide perspective, not all mosquitoes respond to insecticides the same. Well, that's also true for pathogens. Not all mosquitoes can harbor a pathogen the same way. The point is, when a mosquito takes a blood meal, a female, that pathogen has to enter her midgut, it has to get out of the midgut, it has to either multiply if it's a virus, or it might develop if it's a protozoan parasite. Ultimately, it has to get into the salivary gland in order for that mosquito to be infectious and able to deliver an infectious dose to another host. Here is a cross-section of the business end of a mosquito. And this canal here, that is called the food canal. So that's where blood would be drawn up into the mosquito. But down below that is a small structure called a hypopharynx. It's created by the hypopharynx. It's called the salivary canal or salivary duct. So prior to blood feeding, the mosquito actually delivers saliva into the host to just as an anticoagulant to help with that drawing up of the blood. But in doing so, that's when they're delivering these pathogens to the host. So that's how that works. And then, and actually I'll back up. Vector competence for mosquitoes is often evaluated in a laboratory experimental situation. So we can take a variety of species for a particular parasite and evaluate how good they are at the pathogen getting out into the body. So how well does it disseminate? And then what's the level of infectiousness, or how many of them give an infectious dose to another host in a laboratory environment. So that's done. So on the same notion, we also need to pay attention to vertebrate host competence. So which vertebrate host, whether it's a bird, a mammal, or a reptile, they all respond very differently to these various pathogens. In some cases, a particular vertebrate species might actually be a good reservoir. The pathogen can replicate to a high level in their blood, and then they're able to contribute to future transmission. This is a graph for birds referring to this concept of competence for how birds respond when they're infected with West Nile virus. And again, this is done in an experimental setting. It's not always ideal, but that's kind of sometimes our only option. And this can be done for West Nile. We can infect birds experimentally, evaluate how they respond. Obviously, that can't be done as easily for a pathogen where humans are the main reservoir. So this is for West Nile virus. This axis here is a quantification of virus in the blood. So it's the number of plaque-forming units per quantity of blood of virus. And this is the days post-infection here. So the point is, when a bird is infected on day 1, some species of birds produce a high viremia. The virus in their blood gets to a high level. And so that is true for some of the passerines, the perching birds. robins, house sparrows, etc. But for other species of birds like chickens, ducks, geese, they don't tend to produce a very high viremia. And so this is all occurring over a rather short time period. Within a week or so, these birds are either going to die at this time point, or they're going to produce antibodies and recover to this infection. So what's really important is this magnitude of viremia. How high does it get, and how long does it remain infectious? And so there's an approximate threshold. It's not a perfect, you can't pinpoint an exact titer of virus in the blood. But there's a point at which the titer has to get to a high enough level in order for that host to then reinfect another mosquito. So when another mosquito feeds on a bird that has a titer up here above, say, 6 plaque-forming units, that is going to infect that mosquito. So in other words, that host then is going to allow that virus to then continue on into reinfecting future mosquitoes. Whereas many birds and most mammals, when they don't produce a high viremia, that's why they're called dead-end hosts. They're not going to contribute, but that virus really can't get out of that host in any other way. All right. And then I want to make the point that we need to not only be conscious of vector competence and host competence, but then also vector and host contact. We need to know which mosquitoes are feeding on which host, because that's then also going to shape this whole transmission cycle. All right, so speaking of that, again, we're seeing the West Nile transmission cycle. Just a different diagram here. And again, for West Nile, we're looking at this enzootic cycle, this mosquito-bird cycle. And that is simply because birds are reservoirs. They are the ones that produce a high viremia in their blood. And that's why we're saying that it spills over into mammals, including humans. Occasionally, a virus-positive mosquito feeds on humans or mammals, but that is a dead-end route. The virus then can't get back into reinfecting other mosquitoes. So this is the enzootic cycle, that's sometimes called, and often it's termed a bridge vector of some kind, is then bridging that pathogen out into humans. There's a number of potential species of mosquitoes that could be involved, but Helix mosquitoes occasionally feed on humans. So Helix mosquitoes can not only be the amplification vector, but they might also be involved in being a bridge vector. So just so we can compare, here is dengue virus. Dengue virus is also, it's in the same family as West Nile virus, so it's closely related, family Flaviviridae. But it's just a completely different transmission cycle. We're talking Aedes aegypti, so a very different species of mosquito that primarily feeds on mammals, and ideally, from its perspective, humans. And for dengue, humans are the amplification host. Humans produce a titer that is infectious to other mosquitoes. So it has a relatively simple cycle. Mosquito, human, and then that's how this cycle is maintained. So again, from this management perspective, in terms of how we approach these disease systems, can be very different. So for example, a vaccine, people have been trying to develop a dengue vaccine in humans for decades, and it's been really challenging because there's 4 serotypes, and that makes it hard to achieve an effective vaccine. But if a vaccine could be developed, it could be really successful, because the point here is, if you treat a certain proportion of the population, or if you vaccinate them, say it's only 50%, so that not only protects those individual people that were protected by the vaccination, but that could also break this entire transmission cycle. You could actually eradicate and stop transmission. And that's how yellow fever was successfully eradicated from a lot of places. There's an effective yellow fever vaccine for humans. But for West Nile, imagine, what if we did develop a human vaccine? That would protect the humans, just like horses are vaccinated in a trivalent vaccine that protects against several viruses. But if we did develop a human vaccine, the virus transmission cycle would continue. It would still be in the environment. It's just those individuals that were vaccinated might be protected. All right. So again, as a recap, Helix species mosquitoes are considered competent vectors. These species of mosquitoes, they happen to feed on birds. They have a preference for feeding on birds. It's called ornithophilic versus mammophilic. And so that is just by nature. That's what these particular mosquitoes like to do, and that's why they're really good at this enzootic cycle. Culex species mosquitoes, and so for that reason, and kind of as a recap, that's why they're involved in this amplification. And then potentially Culex mosquitoes or other mosquitoes could feed on a bird and then bridge that pathogen over into these mammals. Culex species mosquitoes, in terms of their behavior, we already saw this in one of Janet's slides. They do tend to feed at night. And if you look at host-seeking activity, sometimes there's a peak during the dusk and dawn. But in general, we could say that these Culex mosquitoes are active, trying to find a blood meal all night long. That's relevant here in a few slides. All right, so here is human disease of West Nile across the United States. This is the higher than expected incidence of human cases. And I don't want to pay too much attention to the patterns here. The point is, I want to emphasize something that Janet was talking about. Depending on where you are in the U.S., you have West Nile virus driven by different species of Culex mosquitoes. And so I'm just going to throw the three main groups of Culex mosquitoes that would be responsible for West Nile virus transmission, depending on where you're at. So up in this region, in the United States, kind of more northern and eastern, Culex pipiens, the northern house mosquito, would be the primary vector, the primary enzootic vector. And as you shift south, you start getting into Culex fasciatus, that is the southern house mosquito. It's very similar in a lot of ways. Morphologically, the behavior is very similar. And so in a lot of ways, this West Nile transmission in this eastern half of the U.S. is fairly similar. It tends to be this urban phenomenon. These are container breeding mosquitoes that thrive in this urban landscape, and they feed on birds. They both feed on birds, and so in a lot of ways that's similar. However, Janet referred to Culex tarsalis. As you start drifting west, say in central Iowa, Texas Panhandle, that's where you start getting into the landscape that is more dominated by Culex tarsalis, and then West Nile is transmitted primarily by Culex tarsalis. And so that particular species is very different in a lot of ways than Culex pipiens. It tends to be more of a rural breeding mosquito. Different birds would be involved in the transmission cycle. And then subsequently, if you then calculate human disease, the people that are exposed in these more rural environments, there's fewer people living there. So that's kind of why you see this really elevated incidence of West Nile. So I wanted to make that point. It is not that straightforward. There are Culex pipiens and Culex quinques coexisting with tarsalis, so it's really hard to just simplify it too much. But this is broadly a pattern that is something to be conscious of, especially even it's relevant to right here in Texas, where as you start getting to the Panhandle, you can have these different systems. All right, so Culex fasciatus. Container breeding mosquito, we've already seen a lot of images of the type of stagnant water that they like to thrive in. They're going to be pretty opportunistic. They're going to take advantage of any kind of environment they can find, and the underground environments are probably contributing a lot of production. So inside of any kind of wastewater management system, you've got storm drains. Those storm drains have sumps that are designed to catch all that organic material before it clogs up the pipes. Certainly those sumps could provide container habitat, but then what about all the other underground network of pipes and tanks that could potentially harbor mosquitoes? And it's very remarkable how these mosquitoes can really get into these tiny little cracks. This is a classic grid over a catch basin, so that's easy for mosquitoes to get in and out of. But imagine you've seen these lids for catch basins or even some of the septic systems, where all it is a tiny little hole to put that crowbar in. Mosquitoes can go right in and out of that hole. They can just crank, so larval habitat underneath that environment can still crank out mosquitoes. So that's why they're kind of cryptic in a lot of ways, these environments. But different types of stagnant water, neglected pools, containers, etc., they're going to be pretty opportunistic. And again, they're feeding on just bacteria, algae. We've talked about why some pools are good at producing mosquitoes, why and others are not. algae can be a good, valuable resource for mosquitoes, but some types of algae aren't really toxic. And so that might be one of the explanations for why certain pools tend to produce mosquitoes and others may not. So one general concept is that these Culex container breeding mosquitoes tend to do well in hot and dry environments. Obviously, they do need some amount of water. And so looking back at that 2011 year here in Texas that was incredibly dry, that might have been a scenario where it was so dry, container habitat may not have existed. But if you have a little bit of rain to seed these containers with water, then you might have a scenario where hot weather could certainly drive Culex production. And so the hotter it is, the faster the larval development period occurs. And the hotter it is, the gonotrophic period, that period I was saying between blood feeding events, that can shrink. And also this term that I didn't refer to before, the extrinsic incubation period, is the time from when a mosquito, a female mosquito, takes in that virus. The virus then disseminates through the rest of that mosquito, eventually reaches the salivary glands, and once she's ready to reinfect another host, that's the extrinsic incubation period. And for Culex mosquitoes, that might be roughly 15 to 20 days, so it's actually surprisingly long. It takes a long time actually for that virus to get through these female mosquitoes for them to be ready to reinfect. So kind of by definition, that means it's those old female mosquitoes. They're going to be 15, 20 plus days old when they're actually infectious. And so this is a little diagram explaining this. This axis here, the y-axis shows the number of days, and this is the temperature. So as temperature goes up, the extrinsic incubation period is going down, and the gonotrophic period is going down. So that's generally why July, August is really hot. We see a lot of activity. Yeah?
And how does the virus make it to the salivary gland? Is it just sloppy eating, or is it...
No, it's... So the virus is entering, and it's actually multiplying. So it's replicating. And so as it replicates, it becomes systemically infected throughout the whole mosquito. The legs would have virus, and sometimes there's something called a barrier, a salivary gland barrier. The point is, some species that are considered non-competent for West Nile, the virus cannot get into the salivary gland. And I don't know exactly why that is, but for Culex, when a Culex mosquito becomes systemically infected, eventually that virus gets into the salivary gland, and then the saliva would contain viral particles, and that's how it's getting into the host. Yeah?
And how long did you say that generally took?
About 15 days. In fact, if we look at here's the extrinsic incubation period for Culex mosquitoes. There's 10 days, there's 20 days, so it depends on the temperature, but for an average, so that's 25 degrees, that's around 10 to 15 days or so. So then if you think of this gonotrophic period, the extrinsic incubation period, you're looking at a female mosquito. She needs to be close to 15 or 20 days old before she's even able to give an infectious dose. But that mosquito could test RT-PCR positive for West Nile really quickly. That doesn't mean that this mosquito is infectious, could deliver an infectious dose. And that's also true for, so imagine you could have a non-competent mosquito species, one that has a midgut barrier where the virus can't get out, or a salivary gland barrier where the virus can't get into the salivary gland. Those mosquitoes can still test positive for West Nile virus with RT-PCR or even virus isolation, but that doesn't mean they're an infectious mosquito. So that's why we refer to this the competence. How competent are they fully from getting infected and then reinfecting another host? All right, so I've talked about vector competence, host competence, and then this vector-host contact is really important. So a tool that I used during my dissertation studying West Nile virus is the mosquito blood meal analysis. So that is a tool that medical entomologists use to understand vector-host contact. And now I'm using this same tool for tick systems, for Chagas disease vectored by kissing bugs. So in general, medical entomologists use this a lot for a lot of different vector-borne diseases. And I won't go into the molecular techniques here, but basically the point is you can collect these blood-fed mosquitoes in the field and do a series of molecular techniques that essentially give you species-specific confirmation. You know exactly what species of host that mosquito fed are. And that can be a very valuable tool to gain insight into this whole transmission cycle and identify which mosquitoes are coming into contact with which hosts. So by doing that, for Chicago data, combining the rate at which Helix mosquitoes contact different bird hosts, and then using the competence data coming out of the experimental infection studies, understanding what's the viremic titer, the duration and magnitude, we can kind of consolidate that and determine this force of infection, amplification fraction. It's been termed a few different things. The point is, it's the relative contribution of these different birds to West Nile virus transmission. So how many infected mosquitoes are going to result from feeding on each one of these bird hosts? So for West Nile virus in Chicago, #1 bird here, according to this analysis, is robin, and then house sparrow, house finch, northern cardinal. Typical urban birds, the Helix mosquitoes, they like to feed on those birds, and those birds happen to be good reservoirs for the virus. Now, that's for Chicago. I used the blood meal analysis data coming out of Harris County, and I did the same analysis for Harris County, and it turns out doves ranked #1, then house sparrows, then house finch, and then a robin. So not that different, really, from the data coming out of Chicago in terms of which birds are really driving West Nile virus transmission. And the reason for doves being really high here is based on that data, where Rudy Bueno and others would have collected blood-thin mosquitoes. A lot of them were feeding on doves, and so I don't know exactly why. Obviously, it depends on availability. Yeah.
That's what I was going to ask about it. Because the population of birds, or should that be?
So yeah, the question is about what's the population of birds. So certainly, it's the availability of birds that's mosquitoes. Mosquitoes are not going to be able to feed on a species of bird that just doesn't exist in that area. And so relative availability certainly influences which birds are going to be fed upon. And this is all occurring at night as well. And so in a moment, I'll start to explain how, where on the landscape this vector and host contact is occurring.
Can I ask a question?
Yeah.
Something on that a little bit?
Yeah.
Okay, so since someone made a bad face at me, I almost forgot.
Yeah.
So with this transmission of the availability of birds, since we don't have, of course, your cool lab, would a layman potentially be able to look at the availability of birds during those high temp events or seasons, and be able to just see what, based on how birds fly, where they are,
Right.
and just assume and just start sentinel surveillance based on those kinds of birds?
Right. So it starts to get tricky, and I've been doing this for 8 years in Chicago, studying the birds and mosquitoes. And one thing is, traditionally we did bird point counts, right? So we surveyed the bird community. That is our technicians going out early in the morning, doing point counts. Seeing birds, recording them, hearing birds, recording them. And it's a measure of the bird community. But what I've slowly realized is that the bird community that we detect and see at 8 a.m. or 9 a.m. may not be the bird community that is relevant to these host-seeking mosquitoes that are out there at night. So I'm going to show you some slides about our field operation in Chicago, and essentially our field operation became nocturnal. The point is, at night, that's when the action is occurring. That's when the mosquitoes are contacting the birds. So that's why we want to understand bird distribution and movement at night. That's not a very easy thing to do. That would not be a practical operation for a control program. But the mosquito blood meal analysis can be achieved. So that is a direct measure of seeing which mosquitoes are feeding on which birds. So that can be done, and that is a powerful tool. All right, so again, feeding off of that, the question now is, all right, on the landscape, we've got virus amplification. We know it's these host-seeking Helix mosquitoes. They feed at night. Where are these birds at night? And if we look at this landscape with virus activity, where is the virus amplifying? Is it the neighborhood? Is it riparian areas, etc.? And at this point, I think there are certain scenarios where you could have a source for virus, you actually have areas in the landscape that would be a sink for the virus. So that would obviously be important to understand if we're trying to manage this system. So that's what our West Nile virus project, this is funded by the NSF in Chicago, we started in 2005, but our second phase really focused on this fine scale variation, started in 2009. So the point is we wanted to really understand this heterogeneity in the bird movement and distribution in comparison to mosquito movement and distribution. So a tool we use to study bird movement and distribution is radiotelemetry. And that is a traditional ornithological technique. You slap a little transmitter on the bird, and then using an antenna, you can track where that bird goes. And so again, where these birds spend their daytime may not matter, because they travel a long ways from where they roost to where they feed. And so it's really where they roost for Culex mosquitoes. That's where the action is really taking place. So this is just some radiotelemetry data. I think we did telemetry from 2009 to 2012. We had 191 individual robins tagged with transmitters. We got over 3,000 individual points, over 1,000 of which occurred at night. So the nighttime location, this is us driving around trucks with antennas at night trying to track these birds. And a lot of times it's like searching for a needle in a haystack, because your range is only about a mile as far as how well you can pick up a signal. So these little blue dots that are kind of hard to see, they're all nighttime roosting locations. So robins roost anywhere from an individual bird in a neighborhood, backyard, or robins, and then house sparrows as well, have large communal roosts. The robin communal roosts, the largest ones in our study region, were over 20,000 individual robins. We're all dumping into these areas at night, roosting there. And for house sparrows, some of our largest house sparrow roosts might be 100 individuals. So the point is, across this spectrum, of robins roosting, anywhere from the individual bird to small groups to really massive roosts, at some point across that spectrum, virus is being amplified. We know that's the case. So the point is it occurring at these smaller roosts, or is it occurring at these really large roosts? And we have a lot of data right now that is in various stages of manuscript preparation. I think, though, our current conclusions are that it's going to be these smaller roosts that are occurring like all over the residential landscape. That's the really focal sources of virus amplification. And I actually think that some of these larger roosts are actually a sink for virus. Okay, so to complement the bird movement study, we were also studying mosquito Culex movement. So we used this novel technique, kind of a new tool to study mosquito dispersal. It's using stable isotopes, and I don't need to go into detail. The point is, though, I was able to pour potassium nitrate that was enriched with a particular type of nitrogen, a rare form of nitrogen. Those mosquitoes that were breeding in those catch basins incorporated that unique material into their structural tissues. They emerged, they flew away. This would have been the area that I would have treated in one area. And then this grid of trap, each small little dot represents a trap. And the large dots that are colored, blue or gravid traps are orange, those are where we captured marked mosquitoes. Those marked mosquitoes would have come from the catch basins and that little star, they would have traveled to these different areas. So just looking at this data, this is from 2010 and 2011, you can see that mosquitoes stayed within about 1 kilometer, but there was at least one female that made 2.4 kilometers away. So they certainly can travel, especially when we're looking at 15, 20, 25-day-old female mosquitoes. Certainly there is some level of wind-facilitated travel. All right, so now back to this same study region. We're learning bird distribution, roosting activities at night. We're studying virus activity in the mosquitoes. 2010 was a really hot year for us in Chicago. This map is showing mosquito infection rate for Culex at these different traps. The larger the dot, the more virus activity. And then we also have data showing geocoded addresses of humans getting infected, and there's quite a few of them, humans getting infected right here in this study region. So again, the conclusions at this point is, our thought is that the virus activity somewhat appears random, and that's kind of because we're thinking that a lot of the vector-host contact is occurring in these residential areas, street trees, backyard trees, maybe the small creeks that run through a neighborhood. But those are the focal points of production for the virus, is what we're kind of thinking at this point. So now looking here at the Dallas landscape with the same concept in mind. Obviously, I haven't spent much time here, but I think learning everything I have in Chicago, I think we can use some of those same lessons and apply them here. If somebody were to ask me, where on this landscape is the West Nile virus amplifying? Well, it may not be a very satisfying response, but it's probably just the typical residential environment. That residential environment has the Culex mosquitoes, and it has the roosts, small roosts of different species of these passerine birds, and that's probably a focal source of production. I think a lot of times it's really, again, people associate mosquitoes with water. I think the easy thing would be to say, oh, it must be the lakes, or it must be the larger wetlands that are producing mosquitoes. That's where the birds are. That's where the virus is amplifying and spilling over into residential areas. I wouldn't necessarily say that at this point in my thought process. And indeed, if we look at the distribution of human cases or positive mosquito pools, we see it. It's all over here looking at 2012 data. So the hypothesis that it's occurring in residential environments, it could be accurate. But one thing I wanted to point out as we're interpreting these data going into 2013, this particular map just shows human cases, and that's not that's kind of ignoring the incidence or the number of people living in these different areas. So we certainly want to measure, ideally, an age-adjusted incidence of West Nile virus in humans to see an accurate map showing the relative distribution of where humans are getting infected. Same thing for the mosquito pools. This is just the positive traps. Ideally, we want to see the traps that were negative, or of those positive traps, how many individuals were collected and tested. We want to be able to measure an incidence or prevalence rate to then adequately describe the heterogeneity of transmission in this landscape. All right, so that's where I'll leave off. If there are questions, we can, if we have time.
Yeah.
Okay. I'll start over here, yeah.
How long can that take the larval incubation?
So you're asking about the aquatic stage for overwintering. So for some species of mosquito, they can overwinter as larvae. But, that would be months, yeah. But not for Culex mosquitoes. Culex mosquitoes overwinter as the adult stage, exclusively. So they do not use the egg stage or the larval stage. Yeah, so down here, we could have Culex, because it's still mild, we could have some Culex activity for a much longer period of time. But in Chicago, when it gets cold, there's no adult mosquito activity going on. There's no active breeding. But down here, there could still be. But if you're seeing larvae under ice, yeah, we would want to know which species is that. Could that be one of the species of mosquito that does overwinter during the larval stage? Yeah. Yeah.
Transmission is still limited to humans, that's it, no other domestic animals?
So, yeah, other mammals definitely get exposed to West Nile virus. In general, though, most mammals would not be all that susceptible, but they are going to get exposed. They might get sick, but they're typically going to produce antibodies and recover. So there's been a lot of work looking at serosurveys, so taking blood samples from all kinds of mammals, raccoons, dogs, etc., and they have antibodies to West Nile virus.
We get asked by the park all the time, do the dogs and cats get it?
Right. Yeah.
Yeah, I have a question. How come the virus effect in human population is so much higher than it's not in these United States setbacks?
Right. So the question is basically why was there such a big year in 2012
Contrasting, going back
to the previous year? So yeah, that's the big question.
to
I gave a talk previously about all the potential hypotheses that might contribute to why we saw such a unique event in 2012. I think it's a really complicated set of potential factors, but one kind of simple explanation maybe explaining what we saw was just a unique chain of events. We went for a couple years here in Texas without that much virus activity. Then all of a sudden, the stars aligned. We had a mild winter. We had a relatively wet spring, a dry, hot summer, and then just for whatever reason, the combination of we had a lot of susceptible birds out there. And we had a lot of Culex mosquitoes, and that drove the transmission cycle. That elevated the infection in the mosquitoes, and then the pathogen spilled out into humans. Yeah. Herd immunity is only going to be relevant. The point is, yeah, I have a manuscript we're about ready to submit related to herd immunity in this West Nile system. It's not going to have that big of an impact. The point is, birds have really high level of recruitment. So every year, birds are cranking out juvenile birds. And all those juvenile birds are going to be susceptible. And so even if there is herd immunity, that would only be the adult birds entering the system the year. So, but yeah, you know, if we did not have that much virus activity in 2010 and 2011, then all those adult birds entering 2012 would have been susceptible. They would not have had antibodies. And then all those juvenile birds that year that were born would have been susceptible. So that would have then increased the number of susceptible hosts on the landscape, and that could have really fueled the amplification. Yeah?
Two questions. The assumption is that when we do it overwintering, if it's effective when it goes on, it's going to still be effective when it comes out.
Right, right.
And then secondly, looking at birds, migratory birds versus birds that aren't migratory, is there any work done as far as, like, you had a bad year in Chicago and Kent?
Right.
Those birds come down here as well?
Right, right. So a lot of people have looked into the role of migratory birds in West Nile transmission, and originally we obviously saw this really quick sweep of West Nile across the U.S. And to some degree, birds were probably responsible for that movement. But in terms of, your first comment was about this overwintering mechanism. The point is, how does the virus maintain from one year to the next year? And it's probably more likely to be in these mosquitoes. Like you said, those adult female mosquitoes, a few of them might be infected, and they could remain infected all winter, come out in the spring, and then initiate the new year. Migratory birds, given the time of the year they pass through, typically if it's a migratory bird coming out of, say, breeding up in more north or even in Canada, there's not even that much West Nile transmission going on in that breeding area. So when we catch migratory birds, in general, their seroprevalence rates are really down, meaning they aren't even getting exposed. So I don't really think, at this point, now that West Nile is established, I don't even think migratory birds are really that involved in the system. Yeah?
Have there been other parts of the country that experienced something like we did, where there's not much activity, then a big year, and predictor for this upcoming year, other experience in other parts of the country? Right.
So the point is, now that West Nile has been here for over a decade, we can start looking at these longer-term patterns to see, is there any cyclical pattern? In Chicago, our weather is very, sporadic, kind of like it is here. And so we have not noticed any kind of cyclical behavior. In fact, there have been years in Chicago, like 2005 and 2006, were both big years, back to back. So there could be a scenario where you could have big years back to back. In California, Bill Rising's group has looked at long-term data now, and they see more of a cyclical pattern developing, where once every 3 to 4 years, they're seeing kind of an increased rate. And part of the explanation might be due to this herd immunity concept, where once you have a couple years of low viral activity, then all of a sudden you're going into a season that's primed with a lot of susceptible hosts that could really trigger a big event. So yeah. Yeah.
So can we write off Aedes albopictus as a vector of West Nile virus?
Yeah, anywhere. So questions about albopictus and their role in West Nile. Yeah, in general, they might be considered competent, but in general, these albopictus are not going to feed that frequently on birds. So for that reason, those albopictus are not going to be involved as this primary amplification host. They're not going to be, important in this bird-mosquito cycle. But could these albopictus occasionally spill the virus over by, into humans by feeding on a bird and then maybe feeding on a human? Maybe that occurs. But from a management perspective, it's going to be more efficient to target the human mosquitoes because that is breaking the enzootic cycle. But if you target potential bridge vectors, that's not, the virus is still going to be there. You're trying to target species that would be the bridge vector. I think it'd be more efficient to focus on these kill mosquitoes. Yeah.
Since we get bit a whole lot more by tiger mosquitoes, even though it's not a concern from a public health control West Nile virus perspective, we need to be pretty repellent on being bitten by
Right, so I mean, there's certainly a huge nuisance biting mosquito. Albopictus now swept from the East Coast. Now it's set up shop in California. It's certainly a nuisance biting mosquito. When albopictus first showed up as an invasive species and swept throughout the United States, the big concern was how is albopictus going to be involved in the transmission of vector-borne diseases. And so far, it just doesn't seem to be playing that big of a role. But I wouldn't necessarily ignore it. I mean, when they are sure, they definitely are feeding on humans a lot. Albopictus is a good vector of chikungunya virus. And chikungunya virus went from Africa to India to Europe. Potentially, that would be a big risk of a new virus being introduced to the United States. If that happens, albopictus is here, and then it could become really important from that perspective.
Before somebody asks another question, let's give Dr. Hamer a